Hello. In this video, I'm going to talk about membrane receptors, G proteins, cyclic AMP, and signal transduction. Before we get started, I'd like to define a few terms. Membrane receptors. Membrane receptors are specialized proteins which help communicate signals between the cell and the outside world. G proteins, also known as guanine nucleotide binding proteins, is a group of proteins that act as switches inside the cells that are involved in transmitting signals from a variety of stimuli outside a cell to the inside of the cell. Cyclic AMP is a ring-shaped form of the compound AMP, which stands for adenosine monophosphate, formed from the adenosine triphosphate by the action of adenylate cyclase. In cellular metabolism, cyclic AMP acts as an intracellular amplifier or secondary messenger of signals derived from hormones or neurotransmitters. Signal transduction is a set of chemical reactions in a cell that occur when a molecule, like a hormone, attaches to a receptor on the cell membrane. The pathway is actually a cascade of biochemical reactions inside the cell that eventually reach the target molecule or reaction. In this process, we will look on a postsynaptic neuron, where these membrane receptors are located. Ligand-gated ion channels are receptors that only open to a specific neurotransmitter. Epinephrine is an example of a neurotransmitter that we will be using today. The little green circle is epinephrine. When the epinephrine binds to the ligand-gated receptor, it then stimulates the receptor to alter its shape now that it's an active receptor. Our green GDP is currently paired with the red alpha unit, orange beta subunit, and yellow gamma subunit, which makes up our G protein. The G protein then bonds to the receptor, which releases the GDP molecule, which is replaced by a GTP molecule. So we see our blue GTP molecule attached to our G protein, and now our green GDP molecule floats away. The G protein then splits into two parts, which contains the alpha subunit and the GTP molecule, and the other containing the beta and gamma subunits. The alpha molecule then binds to the adenylyl cyclase enzyme, causing it to activate. We can see our orange beta and our yellow gamma subunits on the left, and we can see the blue GTP with the red alpha unit binding to the orange adenylyl cyclase enzyme on the right. The adenylyl cyclase then starts to convert ATP molecules into cyclic AMP, which continues to carry the messages through the cell. We have the red alpha unit with the blue GTP on the left, the green ATP in the middle, and our cyclic AMP in the bronzy color on the right, all below our adenylyl cyclase enzyme in the orange. The cyclic AMP will then bind to the protein kinase A, which will then phosphorylize the protein. Our protein kinase A is in purple, and the cyclic AMP are the bronzy-colored molecules attached to the protein kinase. Protein kinase A will then release its own activated subunit, continuing the phosphorylation process to the rest of the cell. We still have our purple protein kinase A and our cyclic AMP in the bronze, and now we can see the activated subunit it released in the orange. The cyclic AMP is then broken down by the enzyme phosphodiderase in order to stop the activation of protein kinase A. As we can see, our phosphodiderase in red is emitting the bronze cyclic AMP, but our purple protein kinase A has changed shape, so the cyclic AMP cannot bind. The purpose of this chain of events was to achieve cellular response in the next chain of events for the following cells. As the cells pass on information to each other, this allows the body to react to stimuli and to achieve homeostasis. Thank you for listening, and I hope this video helps you study for your exam.